Hello everyone, this is Núria Queralt-Rosinach. I work at the Biosemantics group in the Leiden University Medical Center in Leiden, the Netherlands. And I'm here to present our work on modeling quantitative traits for COVID-19 case reports. When the world faced a novel disease like COVID-19 pandemia, also described by Dr. Anna Rath from Orphanet like the most frequent rare disease nowadays because of the similarities of the challenges that a novel disease brought into research, the world faced a great morbidity and great mortality that causes great burden for the society. In order to face this situation, it's imperative to facilitate, to improve the access to patient data in an efficient way, to support healthcare management, and also to speed up research to develop new treatments for this novel disease. So in order to support clinicians, when a patient arrives into the clinical setting, we need to understand how they monitor patients and how they take decisions based on the data that they track. So here I show you a sample of a portion of a section of a case report that are the forms that are used to track, monitor, and to monitor patients. The type of data that is gathered, there are several types of data that is tracked and is gathered, but mainly to make clinical decisions, the main data would be quantitative traits as vital signs like temperature or blood pressure, and also lab measurements like the level of hemoglobin in blood or the count of white blood and also qualitative traits like phenotypes or symptoms. So this data usually is tracked on these forms that usually are in different formats made with different designs. So this is very fragmented data. Case reports are very fragmented data. Why? Because there are several kinds of designs. Case reports we can find, this one is from the World Health Organization, but we can find different designs even in different public institutions, public health institutions, different hospitals. So, and also another problematic are the format. They can be on paper, they can be electronically, and electronically they can be in different systems like Castor, REDCap. So what we need is also to track this information in a standard way and to be accessible not only for humans but also for computers to make this data shareable and also foster coordination and collaboration. So, quantitative traits are valuable for clinical interpretation because they give complementary information to phenotypes. They can inform how the progression or the course of the disease is occurring and can give information about how the prognosis can be or how a patient is responding to a treatment. Excuse me. But also are very valuable because they can give information about the pathways, about the organs, about the mechanisms underlying phenotypes at research level. And also they can be key to bridge the gap between the clinics, the bedside, and the bench side and back. So the problem to interpret this data is to integrate how these measurements, how these clinical measurements are related to phenotypes, to which ones, and how they can support or can help give evidence to clinicians to understand the disease, to understand the phenotypes, to make decisions in the clinics. So how to interpret this data? The current solutions for that are the use of formal ontologies. Formal ontologies are computationally accessible, and during the last decade, it has been a great effort by the life science community to develop ontologies because they help to encode knowledge from different domains, from genotypes to phenotypes. And also, there is a standard, the clinical measurement ontology standard, that encode information about these measurements. However, CMO, the clinical measurement ontology for these quantitative traits, lacks of links to phenotype ontologies. And this is crucial in order to integrate this data and try to make clinical decisions based on that. So, in front of these needs of having machine-readable models for quantitative traits in order to make this data more shareable, and also to link quantitative traits to qualitative traits for clinical decisions, our goals were to make formal descriptions for these links, quantitative to qualitative traits, and also to develop models for machine-readable quantitative traits. Our approach was, this work is a preliminary work, and it was developed during 1 week in the context of the biohackathon devoted to the COVID-19, to fight COVID-19, and is a community effort. So because we were in the ontology group, our approach was to use ontologies and formal descriptions, and we use case report forms because it is where the data is, quantitative data is. So as a first result, there is a workflow that we follow. First step was to look for where is the information, what are the sources for quantitative traits. And here in this stage is where we realized the great fragmentation of this way of reporting cases, disease cases. So we ended up to decide to use the World Health Organization case report form as a standard. Then from here we collect manually quantitative traits. Then we map these traits to existing ontologies in the OVO Foundry. Then we develop formal descriptions using description logics in OWL. Then we applied these models and these formal descriptions to reason and for data sharing. And finally, the strategy was to gather, to store, to make public freely available code and models. So first we mapped these COVID terms to ontologies, and we extracted 57 quantitative trait terms from the WHO case report, but also from immunologists that had experience caring COVID patients. The conclusion was that using CMO, we could map the 70% of terms. And for the units, using the unit ontology, we map the majority of the terms for units, but only the 63%. So if we compare this coverage separately for the terms extracted from the case report and also through immunologists, what we can see is a difference. We can see that CMO decreased coverage when we try to encode terms from that are important for the domain experts. So this highlights the importance to keep in the loop the domain experts to solve the driving use case and to encode important knowledge into ontologies. Then we made this formal linking of quantitative traits to phenotypes. We developed these axioms in OWL. The strategy was to make these descriptions using computable logical definitions for classes in Simio, and these definitions as compositional, so classes as compositional classes, and this was a two-step strategy. The first part is the lexical mining, so we map Simio to other ontologies through the mapping, through matching of labels to the labels. So for example, and we use Pato, Veron, and Kevi to map, to link, to relate measurements to qualities, phenotype qualities, anatomy, and Kevi, and molecular at molecular level. So, for example, the class blood pressure the blood pressure measurement in Simio can be decomposed with Pato for pressure and Uberon for blood. Then we formalize patterns based on the EQ description, the entity quality description, that is a description used in describing phenotypes in phenotype ontologies. And once we had done these patterns for Simio classes, we integrate these patterns with these axioms in OWL with axioms in Simio, Kevi, Uberon, Pato, and mammalian phenotype ontology. So, as an example of these axiom patterns, we can see that blood pressure measurement can be described with this axiom using these classes and linking Simio to Pato and Uberon. And all this integration was stored in this OWL file. Then we can apply these axioms to reason. For reasoning, we can apply it to integrate these quantitative traits to phenotypes using reasoners. So we can infer relationships between measurements and phenotypes that may be detected using these measurements. So we can use this inference for interpretation. For example, we can relate these measurements of the concentration of immunoglobulin A to immune system phenotypes. Next, we model quantitative traits, mainly using we develop a very simple model, just using 4 concepts, using the measurement described by the clinical measurement ontology, using the assay that it was used for the measurement, using OBI, using ontology biomedical investigations. Then we can also define the value attached to this quantitative trait, to this measurement, and also the units using the unit ontology. And also we defined these measurements using the information artifact ontology. So with this model, we can transform data to fair data. And also we implemented these shapes. This is a language that allows us to communicate graph structures and also to validate instance and to validate data and the model per se. So then we applied this model to extend this standard for sharing phenotypic patient information in the clinical settings and in research, a standard that is called PhenoPacket, and it's a standard of the Global Alliance for Global Health. and it integrates this patient phenotypic information, but also what is very interesting that it allows to integrate genomic information. So we extended this standard, developing a new schema based on the model that I've just shown for quantitative trait futures, and this allows us to attach clinical measurements to the packet schema for data sharing. So what we have developed are formal descriptions to support data management and interpretation for research and in the clinical settings. Also, we have made Simio more interpretable with Uber and Kebi, Patwa, and phenotype ontologies. Also, we made these quantitative traits computable and enabling inference for integrating this information that may facilitate to make connections between measurements and phenotypes. Also, the models are useful for data sharing, but we have these limitations. With a unique ontology, we cannot describe all the terms, and we only base this study with one form, so it's not comprehensive. So, in summary, our contributions are the integration of these measurements to phenotypes for clinical interpretation and biomedical research. To make this, we have developed these fair machine readable quantitative trait models for data sharing. That this is a community effort. Very wonderful because we work with people that we don't know each other actually. And then also we made this code and models freely available. So in summary, what is important here is that we have made more accessible these clinical measurements, not only at human level, but also computationally, because we now can describe in a formal way these measurements and making them linkable and combine this data with research data or other data. And also, we have made this information linkable with other with other knowledge domains, more linkable, and that helps interpretation. So finally, I would like to thank the COVID Virtual Biohackathon organizers and also the Ontology group for all the wonderful discussions. I learned a lot. Also, I'm very grateful with the Biosemantics group for all the support and good vibes. Also, because I developed this work 1 week before starting working with them, so it's very nice that they allow me to develop this. Also with the Loom clinicians, lab manager, researchers, because I'm involved in the internal COVID project, and it gives me the opportunity to understand a lot the needs and requirements. Also, who funded that I'm here, the European Joint Project for Rare Diseases, and finally the ISMB organizers. So thank you all, and I'm open for your questions, please.